to this online workshop on data science for biologists. Data science is always center of interest to researchers. This workshop is jointly organized by Amity University, Rajasthan, Jaipur and Pine Biotechnology. I would like to welcome Dr. Mohit and his team from Pine Biotech and Professor Binay Sharma and Professor Neelam Jain, ma'am, from Amity University, Rajasthan, Jaipur. As well as I also heartiest welcome to all participants to this online workshop. Now, I am happy to invite Professor Binay Sharma sir, Dean Research and Director, Amity Institute of Biotechnology, Amity University, Rajasthan, Jaipur. He has decades-long experience in teaching and research as well as in administration. Sir has diverse experience in various domains of life science research, including biotechnology, bioinformatics, and plant sciences. Sir has more than 400 publications with more than 6,000 citations and 40 H-index and 150 I-10 index. Many prestigious awards and recognition are associated to his name. Now, without taking much of time, I would like to invite Professor Binay Sharma sir to deliver his insight on workshop theme. Sir, please.
All right. Thank you. Thanks a lot, Dr. Ravi, for this, I think, organizing this very wonderful session, this workshop on data science. And first of all, at the outset, once again, on my own behalf as in capacity of Director, AIB and Dean Research, and also on behalf of the Department of Biotechnology, the Amity Institute of Biotechnology, I would extend my very, very cordial welcome to our esteemed expert guest, Dr. Mohit Mazumdar. Now, in fact, Dr. Ravi used the word insight. Now, incidentally, I do not really happen to be, an expert on data science. And I would rather say I would give some introductory remarks on the data science. But I've seen it, I have been I am very closely interested in the area of bioinformatics. And I handle the Department of Biotechnology, Government of India, sponsored Center for Bioinformatics for a long time, in fact, almost for 20 years. So I have seen that how this area has grown and how this domain has come more and more in the area of biological sciences, how it is occupying greater and greater importance, and rapidly so in this field. In fact, now we cannot really think of doing anything worthwhile in biological sciences without the use of bioinformatic tools. Now, let me very quickly share with you a few slides which will introduce you to this topic, and later, of course, you are going to experience the exciting have real hands-on sessions, including the scientific games. So I'm sure that you are highly excited and you look forward to that session. But before that, let me share a few basics of the data science. You have to understand that first, because I know that we have these students from all backgrounds, and let me make them at par level so that they go further into the specialized field. So let me share the screen. All right, I hope, can any of you please confirm that you are able to see the screen?
Yes.
All right, thanks a lot. Okay. So my first worry is of course happens to be in reference of biology. Biology as a data-driven science. Now, in fact, we have gradually progressed a lot in the area of biological sciences in terms of data. The data in biological science is growing at the exponential rate. You know the power of the high-end DNA sequencing. In fact, there was a time I remember that 1989 or so, we had a target of 15 years for the Human Genome Project. And in 2000, it was ahead of time. 2000, we have, uh, the draft sequence of the human genome. In 2003, we had the finished sequence. So it used to take so much of time. It was a huge effort. Millions of billions of dollars were thrown into it. It was a multi-country participation. I remember that as many as 15 countries participated in this effort to sequence the human genome. And now, it makes really us wonder that these days you can get the entire human genome sequenced in a span of about close to 30 minutes, 40 minutes, something like that. So that is how we have grown in this area. You can imagine. So this is to give you an idea that at what rates the data is exponentially growing in the area of biological sciences. Just one example was that of the DNA sequence. We also have, the protein sequence data. And data, this happens to be the massive data, proteins and the nucleic acid. But other than that, in virtually all spheres, we have huge data, which is available for interpretation, which is available for predictions, etc. We have mass spectrometers, which are used in a very big way for the peptide data and many other kinds of data. We have the very high-end digital microscopy, which as well, the data of that as well needs to be interpreted. So we have to understand that now the data science has come to not only stay in biological sciences, but has acquired huge importance here. Let's move to the next. And now what kind of science is the data science? In fact, it is highly multidisciplinary science. In fact, you are very aware, if we talk of the bioinformatics as well happen to be multidisciplinary. In fact, biotechnology itself is a multidisciplinary science. It requires inputs from many different areas of not only the biological sciences, but also the other branches, physics, chemistry, and so on and so forth. But data science happens to be highly multidisciplinary. So first of all, you have to have the domain knowledge. Without domain expert knowledge, you cannot really do anything. Because you will be at a loss to understand what you really need to do. So as you have to be a domain expert, you have to acquire sufficient knowledge that be it molecular biology, genetics, biochemistry, or biophysics, as well as many other fields. But they happen to be the main. Then one has to have a suitable background in mathematics and statistics, because they are the core of the computing, the computational sciences. Because without its knowledge, sufficient good background knowledge in statistics and mathematics, you are not going to develop algorithm, or you are not going to know that how to manage the data, etc. So then comes the third part, is the computational part, where you develop the algorithm, you manage that kind of data, use data. And these days, in medical biology in particular, this image analysis is developing rapidly. Major advances have taken place. So mainly three areas in the computational biology. But let's say if we talk of the history of this, that how did it start? You might really wonder that in early 1800s, we had the instances of biological, what we call as the big data. At that time, it was called as quantitative biology, which essentially is the precursor of the modern data biology, biological data science these days. So Gregor Mendel, way back in, let's say, 1850s or so, he studied, all of us as students of biology, we know the Gregor Mendel's contribution. He's called as the father of genetics. So he studied 7 P traits or characters. And then based on this, he established the principles of inheritance. And he had as many as 30,000 pea plants. So 30,000, I repeat, not few, not 100, but 30,000 pea plants. So it was the beginning of the quantitative biology. And then some of these, then based on the studies of these 30,000 plants and the traits which were involved, he established certain laws of inheritance. And but these requires the help of the mathematical tools, and you know uh, he recognized certain patterns based on this, and then he demonstrated that all these patterns not only not only apply to P, but they also apply generally to the biological living organisms as a whole. So that was as early as 1850s or so, quantitative biology. But then, let's say for a long time, I would say at least 1970s or so, the biological scientists were not so much crunched for the needs of the data science. But gradually it has happened, over a period of time. We have talked about it. We have an expanding collection of sequences going on now available in public domain also. And there are many genome sequencing projects, and TIGR happens to be the main database for the genome sequencing projects. So we have expanding collections of sequences, and how these the raw sequence doesn't really convey much. They don't make sense unless and until you analyze them, you interpret them. And with this kind of data, when I said that 3 billion sequences now being sequenced within less than an hour, then is not an ordinary thing. IOU cannot do it, human effort cannot do it, ordinary computers cannot do it. So you require high-speed digital computers for this. Essentially, the genesis of the high-speed digital computers were the weapons research program, which was developed during the Second World War in 1940s or so. And the weapon research programs, the computers which were which were used in the weapons research program, they were eventually made available to the academic biologist. So you see, if we see how it has grown, if we have a look at the history, the major events, forget about 1850s or so, but let's say begin from 2000. So 2000, we know that, we used to have some kind of cards at that time. In fact, cards we used to have sometimes, I would say 70s and 1980s. And these cards could store a lot of data. But in fact, I have also not really used them, and they were, they belong to the time, not of my time also. But gradually around that time, we started to have, as you know, and many of you are aware also, that we started to have the discs, CDs. So CDs, the cheap discs were available, which IOU could buy. We also have the hard disk, which could, be attached to the computer in order to, increase the space, the storage space, and they also had huge powers, and they are continuing to be used now. So we had these cheap discs, and then gradually they have grown. We had CDs of this capacity, 540, then gradually so many MBs, and they have also grown in their storage capacity. CDs, we had DVDs, and after DVDs, you had, these, pen drives, which have come. In fact, now the CDs and DVDs are also forgotten. So we have seen it evolving, So if we talk of the data storage has become gradually cheap, if we talk of early 2000s, big development, Hadoop was a open software database which was used. It was collected from the public domain and which was used. And then we had what we called as the deep learning. Deep learning is something where the principle is derived from the biological systems. So you had the development of neural networks, for instance, deep learning, which is essentially a part of the machine learning. We also had other developments. For example, we had something called as Google Flu, where they tried to predict the trends of the flu, but they were not successful, so it was discarded. They were not successful because of many reasons. I may touch upon that a little later also in my talk. So the data science then gradually entered 2010. We had found that it was maturing, and then there were mass methods for dealing with the large data. And then, gradually as we proceed in, let us say, next to 2010, especially to mid 2000, let's say around 2015 or so, we find a lot of things has happened, and AI has, you know, uh, taken in a very, very big way. But then you see, another major development, parallel development also took place. And that development was the need of the biological companies, the pharma companies, and the companies who were dealing with the biological data, their need to realize and and collaborate with the big computer companies, like with big data companies, like for instance IBM. And at that time, it was already realized that the data is growing not only exponentially, but a very, very rapid rate, as 90% of the world's data was created in the preceding 2 years. This was IBM statistics. So there was a need to integrate this with the biological sciences, with biotechnology. And then we saw the collaborations which came, many collaborations which came between the core computer domain companies and the biological sciences companies. Pharma sector, for instance, Pfizer. Pfizer was a big player. It collaborated with Watson. Then we have Roche, GNS, causal machine learning collaborations. Novartis, IBM, Watson collaborations. So these were then very big collaborations, realizing the importance of the data interpretation, the collaboration of the big pharma companies, big biological sciences companies with the computer companies who were dealing with the big data. So we had cheap disk, then comes the big data, then cloud computing. All of you are aware. I think we all are using cloud computing because these Google Drives or the Google Drives or the other many things, new drives, Google Drives, they are storing our data. Even we are using it ordinarily, and then we are getting it from the cloud, and then it is available. We can run it, we can use it, but we don't store it. Storage is done by somewhere else. So then we had cloud computing, mass analytical tools, data scientist team, and leading to all new analytical insights into all this. So there is something called as the value, that what is the value of that data and what is the difficulty in understanding that data. So we had the raw data, it doesn't really make sense. We have the sequences, the DNA sequence, but don't really make sense. So then that is the background. We have that raw data, and as you interpret it, it becomes insight. That is, you are able to interpret the data and draw certain conclusions. And then based on that, you are able to conclude, you are able to predict certain things that can be foresight. So initially, we have only the descriptive analytics, that this sequence, then diagnostics, that do they really mean something? That if you mutate the, let us say, X sequence at a particular place, then what kind of change it is going to bring about? It, of course, had a lot of applications that you are able to diagnose. Diagnostics, I did not emphasize all that, I think. So huge applications. So we are able to, through diagnostics, analytics, we are able to conclude that how it has happened. So this has happened because of the alteration in the sequence at such and such place. And then based on this, you are able to predict. So that is predictive analytics. So all these possibilities exist. So what is going to happen next? And then you finally come to the prescriptive analytics, where the directions are given out of many, let us say, into, they are more specialized, they are more specific, and then what is going to be the final outcome and what measures we should take. What should be our approach in order to solve all those difficulties? So that's the way it has grown, And so essentially, in order to arrive at a decision, we need the cooperation of the descriptive analytics, the diagnostic analytics, the predictive. All three are, let us say, together, they are leading us to arrive at a decision and the action is coming by way of the prescriptive analytics. Based on all the information which we have by descriptive, diagnostics, predictive, we come to the prescriptive analytics where a specific action would be emphasized. So I think it's a kind of a repeat. I emphasize this once again. So what are the components of data science? Domain, knowledge, whatever it is, be it business, be it biological sciences, we are working in biological sciences or sociology. And then the second major component is that you have to derive appropriate metrics for summarizing and normalizing the data before you attempt the computational analysis. So you have to have knowledge of theoretical statistics, applied statistics, research design, network modeling, and then finally computing, programming, algorithm, machine learning, and AI, computer graphics, database, and so on and so forth. So essentially, this I think is almost the end of this slide. Maybe one more of this. So, is it going to be challenging for biological sciences? Definitely yes, because we are aware that biological sciences, they are natural sciences and they are not governed by fixed factors. There are so many variables which come into play. And these variables are also governed by, another. You may have thousands or hundreds of thousands of variables, and these hundreds of thousands of variables may be governed or may be influenced by hundreds of other factors. Now, what we have already seen in the COVID times, there were predictions. You might recall that the computer scientists made predictions, a lot of predictions, that this COVID peak is going to come now, it will come in June, it will come in August, it will come blah, blah, blah. So many things will happen. But they were not correct. They failed. Virtually all of them failed because the peaks did not follow the predictions. The top-notch computer brains of our country, as well as globally, made all these predictions, but it didn't really come to be true. Or at least it was not accurate, accurate at all. Why we failed? We failed because the data, if there were, thousands of variables, we had information only about, let us say, 20, 100, 200 variables. So unless and until you have the information about all the variables, I think the predictions would fail. Predictions, because ultimately the data science will use the data in order to make the predictions. It's going to use the data which is available. If the data which is available is insufficient, is not complete, and more important data is missing, then I think the predictions will not be accurate. So it's going to be very, very complex. But how you are, I will repeat the second one which is written. However, given the size and complexities of these relationships, variables and interrelationships as well, exhaustive analytical pipeline therefore requires an end-to-end integration of approaches, forming an analysis stack, starting with data collection, continuing through computational and statistical evaluations towards high-level biological interpretations and insight. I think that is all. So this is last. So therefore, with the COVID example which I gave you, it is naive. We can conclude it's naive to only want few but correct answers, as you can get blindsided. So with 10 informations, whereas you require 100 informations, you will not be able to, you get blindsided. You will not be able to really conclude anything. So for building robust models, for understanding a phenomena, we need more data. All right, so that is important. More and more data, more and more accurate data, in-depth data regarding all variables. Unless we have that, it is going to be very challenging. You may not be able to draw highly precise and accurate, conclusions from the data. Okay, so what is the key? It's essentially a repeat, but I think it's good to repeat now, so that you are able to recollect. What are the key takeaways? Descriptive, diagnostic, predictive, and prescriptive analysis. They are the four major components of data science. So the first three, they take you to arrive at a decision, and then the fourth level guides you in taking the action. So any level of analytics involves three components: domain knowledge, maths, statistics, and computing. Data science investigation follows the same procedure as the normal wet lab scientific experiment. It doesn't require some technology to be imported from another planet. but essentially you can apply the principles of wet lab biology here as well. And then biological data science acknowledges that computer science, mathematics, physics, statistics, and other the quantitative fields have developed advanced techniques that can be applied towards understanding of biological data. Because if they are not, then the data is so complex that it will lead to faulty conclusions. And therefore, any analysis of massive data will unavoidably generate a certain rate of errors. Good research and development will include an evaluation of error rates, and improved methods only can minimize the error rates. So there is always a trade-off, therefore, between specificity and sensitivity. So I think essentially that is all I thought I need to share with you. And if I can stop sharing, let me find the key for that.
Thank you, sir.
Okay.
Thank you, sir. Yeah.
Okay.
Wonderful
All right.
overview.
So thanks a lot. So I think now the students have the background to, understand the challenges which will be thrown in front of you by our guest speaker and expert in data science, Dr. Mohit Mazumdar. Thanks a lot. That is all from my side.
Thank you.
Thank you, sir, for
Thank you
your
so much.
time and such insightful talk. Now, I would like to invite Professor Neelam Jain from Amity Institute of Biotechnology, Amity University, Jaipur, Rajasthan. Secretary, Industry Advisory Council, Amity Institute of Biotechnology. Ma'am has research interest and vast experience in infectious microbiology and bioremediation. And I must thankful to ma'am for her keen interest in industry related institutional activities. So ma'am, please hand over to Professor Neelam Jain, ma'am.
Thank you, Dr. Ravi. And I must congratulate you to online workshop on data sciences. It's a real good initiative. So good morning, one and all. And I, on behalf of Industry Advisory Council, Amity Institute of Biotechnology, I welcome our industry mentor, Mr. Mohit Mazumdar from Pine Biotech. Dear students, you would be glad to know that Industry Advisory Council is actually comprising of our industry experts and alumni students, and it was seated in 20 with the aim to bridge the gap between industry and academia, and for also mentoring and making our students industry ready. Since its inception, ISC is involved in curriculum development, conducting guest lectures, workshops, conferences, and conclave, industrial visits, and many training programs by our industry experts, so that our students can get a wider exposure what is happening in the industry and biotech particularly. Pine Biotech is actually not new for us, as it has been always a hand-holding for our students since 2018 by conducting various workshops and lectures in the field of biotechnology and bioinformatics and data analysis and management. I therefore urge all the students and faculty members who are participating in the workshop to take active participation in it and be a part that is and which will have a long-lasting impact. Extend a warm welcome to Mr. Mazumdar at AIB family, and thanks for accepting our invitation to conduct this workshop. I again congratulate Dr. Ravi for initiating this workshop, and I look forward for a fruitful interaction ahead and a beautiful session. Have a good day and best wishes to all the participants. Thank you.
Thank you, ma'am. Now, I would like to invite Dr. Mohit Mazumdar, Director, Global Business Development, Pine Biotech. And if I would like to introduce Dr. Mohit in a single word, then he is a dynamic person. So the word dynamic is perfectly suited to him. And He has started his journey from GNU to Accelera to Pine Biotech, and he is heading global position, as well as one more thing about Dr. Mohit, I must include here that he is very friendly person. Whenever I approach him, so I always feel that I'm speaking to one friend, nothing like I'm speaking to a global business development director. So thank you, Dr. Mohit, for accepting our invitation for this workshop. And I request you to first start your talk by introducing Pine Biotech to our participants. And I also welcome Ms. Sonalika from Pine Biotech. I always used to speak to her for planning various activities associated to Pine Biotech as well as Amity University Rajasthan. So welcome Sonalika and other team member from Pine Biotech. Dr. Mohit, hand it over to you.
Thank you, Dr. Ravi. So before I start my session and interact with the participating students, I would like to thank Professor Neelam Jain for this insightful discussion about the industry and academic collaboration, and especially would like to thank Professor Dr. Vinay Sharma for his insights and giving us like this platform that actually really helps me a lot. And I think this is it. I mean, this is very important, that how industry and academia can sync together and provide best of their resources and knowledge so that the students and those who are learning can benefit the most. So I think in terms of academia, there is a lot of knowledge, there is a lot of experience, and with industry, there is some practical aspects that we know about industry that is kind of expected for job opportunities. So combining these two different areas of expertise from two different organization is a great initiative. So thank you so much for inviting me to be a part of this. And I'd like to especially thank Dr. Ravi Ranjan for coordinating and providing this unique opportunity for students through this industry academia collaboration. So with that, I'd like to start the session today, which is more about learning and understanding why we should be doing, where this is going, and also what are the opportunities, as well as where can we start from. So today I stand before you to discuss a field of science that holds incredible promise and potential. This is the world of data science and world of bioinformatics. So this is, as Professor Vinay said, this is not just a subject. This is a gateway to a world where biology and technology converge to create a future that's as exciting as it is impactful. So I wanted to ignite within you the motivation to consider joining the bioinformatics industry, a realm where your skill and passion can truly make a difference. So bioinformatics and data science are kind of similar fields, but there are some key differences that you already have learned. So both these fields hold specific experience requirements, such as knowledge of computer programming, as Sir was saying, machine learning, statistics, data processing, data visualization and transformation. These are very common practices, what we are regularly doing. So experience with some of these primary programming languages that you have learned earlier is kind of useful, or maybe I should say that you being comfortable with the computer is one of the most important thing. And now with the advancements of the technology and mobile phones are being so powerful, I think everyone is comfortable with the interface, with apps and all. So I don't think that it's a big of a challenge right now for this generation. So these are some of the key skill sets or things that we were used to look at before. But in the last 2 decades, since the Human Genome Project was completed, so the cost of the genetic sequencing has dropped from 2.7 billion. So that's how much it took us to, do the Human Genome Project, to less than $100. So you can imagine that how the technology has changed and evolved over the past few years. So simultaneously, you've been hearing about the advancements in technologies such as gene editing or genetic editing techniques such as CRISPR that has made it possible to edit living organisms, to make biological medicines and create new agriculture products. So there are a lot of things that we will be talking about in today's session. But I am really happy that you all are here and going to contribute in this field one way or other in the future. So about the Human Genome Project and all this explosion of data. So this activity, all of this led to this explosion of large amount of genetic data, which is now available available in humans, which means that we have a lot of human data, and as well as other living being data as well. And a growing need for professionalism is required in the background in this data analysis and biology integration. So how can we manage and interpret these data sets using advanced statistical technologies or machine learning technologies or AI is somewhere there is a huge need. So opportunities for high paying and rewarding bioinformatics careers are growing. And I know this for a matter of fact because all these jobs that people are these companies are floating research labs and universities. So we maintain a group on LinkedIn where we are posting about these jobs and connecting students with these several types of opportunities. And also a lot of companies are reaching out to us directly because we are a training and research company. So they are reaching out to us for specific type of application that they have, and they are looking for students who can help them and join them as an employee. So there has been a huge growth in this because I started bioinformatics at a very, very early stage in 2008. Since 2008, I've been practicing it, and then before that, I was learning bioinformatics in my master's and undergraduation. So I've seen that how the market has grown and where we were before and where we now. So according to in the US, there are like huge opportunities. People are going, like many of the students who are completing sort of trainings are specific type of trainings are going to US for undergraduate studies, postgraduate studies, postdoctoral studies, PhD scholarships, and all of that. And there are different types of awards that are being awarded by these pharma companies, like Regeneron is one of such company that has a huge computational infrastructure, and they run this science school or science fair, which is very reputed, and any student can participate in that and give the proposal and win a prestigious award and scholarships. So there are many opportunities like that once you have something that you are interested to work on and you have done some work on that. So as I was saying that according to the Bureau of Labor Statistics, job in computer science or computer-based analysis are projected to grow 22% by 2030. So with the healthcare, pharmaceutical, and biological fields leading the way, other fields where bioinformatics plays a critical role, such as microbiology, zoology, and wildlife biology, they are also with the same pace, and they are above national average in terms of growth of salary and other things. But these are all good things to talk about. But to reach here, there are also, it's not such an easy path. So understanding what is crucial here and what is a point to take away, what is your takeaway point would be to understand what are the actual need of institutions and industries, which is that what they are looking in the students. So it is going to be critical that the students who wish to be a part of this generation where you are like a highly paid skilled employee, or you have a startup, or you're running a company, or you're doing research in a cutting edge lab, or you're contributing in drug discovery. So if you want to go into that path, and if you want to help in healthcare sector, I think it is going to be critical for you to understand that what are the actual requirements of the industry, what are the actual requirements of these institutions who are looking for students or interns or employees for these specific types of tasks that they have. So to help in that, that's why we are here in this in this academic industry collaboration to be able to give you those insights and help you so that you can directly be, plugged and play into the industry, and you grow in a very faster rate because you already know what is being expected from you and you kind of deliver those things. So one other thing is when we were listening to Professor Sharma, we learned about that it is an interdisciplinary field, this modern data-driven um, sciences, and it is not just bioinformatics, but every science is now turning into modern data-driven science, modern data-driven approaches that are implemented in these different types of research or sciences. So why is that? I mean, and how to understand this? Like a very simple example is I keep on saying that whenever I buy a product, usually before that, when I was a kid, we used to go to a shop and we will go there and we will look for options that the shopkeeper is being providing us. It could be like before, I'm a Bengali, so we have Durga Puja, and we usually get a lot of new clothes during Puja. So we will go to the shop and we will have limited option, or we'll get from different relatives, and they will provide us with whatever they like. But now what is happening is that these online markets and so many options and so many different websites is providing us with a lot of data. And this lot of data that we are being bombarded with, it's happening all the time, like in social media, Instagram, everywhere. We are looking at a lot of information, and that is making us, things to, it's difficult to choose now. It's difficult to figure out that what would be the right decision because of so many, distractions, I could say, or there are so many options, I could say. Similarly, that is also the case in education. So this modern education is constantly, first of all, there are innovations happening. It's rapidly evolving, and at this time when I'm talking, there are many AI companies who are doing so many different things and showing you saw that what happened with Chat GPT. It just transformed so many things, and we are just at the scratch. So we are not even like reached the potential. We just at the beginning of all of this. So there would be a lot of changes, and that we have to adopt to really quickly. And that is the main kind of would be skill set that you need to have to rapidly change and learn things and implement things as they are needed. So that's what we are trying to do when a new tool comes in. So very recently last year a revolutionary tool came in for structure biology, or structure prediction, which you might have all have heard about, which is AlphaFold. And then it changed a lot of things. So we all had to adapt to it, and we had to see that what are the limitations, how can we maximize its usage. And that's what the industry is looking at. So if you know that tool and if you know how to use it, they will be interested to hire you. Similarly, ChatGPT and large language models are completely transforming the landscape of pharma and all of these different type of biological research and even biological research, like all type of biological and life science research. So how do we get to at par with it and really be relevant for that's how we are going to get the opportunities, right? So one of the challenges, as I said, is the vastness of things. The internet universe is so vast, it's really difficult to understand that where to start from and what would be a good way to start, especially at a beginner level, because there we need the maximum help. Once you are comfortable, once you are an expert, now right now I don't feel like I can learn anything from quantum mechanics to computational doing deep learning. I can put my hand anywhere and try that out because I'm now comfortable. It's more important to become comfortable in this, and that's why I think that's the reason we are all working towards it, to make it comfortable for learners and especially for new generation, new students, so that they can be prepared for the future. So in this session, as I explained, why learning about the modern data-driven is not that simple, I explained because of the vastness. Why practical experience and guidance is required? Because with the experience, we see things differently. We know that this could be the right way because we have gone through it, had years of been working in this area, and now feeling like that we can come up with ways that could really help students. And then why you should do it, like I should ask, like, why, what is the need, is it really needed? What if I'm not comfortable with programming and statistics? Because many of us joins like things are like scared of mathematics, and mathematics and statistics kind of makes a lot of this part of this area a relevant one, because when you're talking about data, and this is a common practice, like even in cricket matches you see like scorecards, and you see data and you see statistics all the time, and that interests you the most, like is how much, like most number of runs, most number of fours, and you see statistics all the time, and statistics is engaging. So you cannot let go statistics and say that I'll not learn statistics because I am scared of it. So those are not the things. So that, I mean, to be able to help you, we that's where like our company comes in and does a lot of work to simplify all of this, so that there is an easy pathway and one can start this journey and then explore on their own. So little bit of characteristics of this data, because we always talk about data, and when we are talking about data, we are thinking about different types of data. But when we're talking about big data, it's not as simple as opening this data in an Excel sheet and doing and seeing and understanding what is in there. Here it's all jumbled up, and it's massive in size. One of the biggest challenges is the size. So this big data, how can you say that a data is a big data, or is a data is like a normal data or small data? There are some characteristics of data that explains the type of data it is. So before you go into and like get into this field, it is important for you to know where you are heading towards. So let's understand this big data. So big data has 5 characteristics. First is volume. The amount of this data is huge. For example, a genomic experiment. So when you're sequencing an organism and you're looking at the genomic information, so you have 3.2 billion base pairs, right? And you get that data in a file. It's a huge file because it has 3.2 billion base pairs, and that's a lot of information. Then you do an RNA-seq experiment, and it yields information about tens and thousands of genes, or millions of single, like in case of variants, it could be a single nucleotide variation. You're looking from millions of points. So how can you figure out which one is relevant? So these are some of the challenges with big data, and that is what we are trying to solve all the time. Second is variety. There is genomics data, there is transcriptomics data, there is clinical data, there is information about age, your weight. So all of these are different types of parameters. So this explains a single person, which is such a complex person. We have such a complex biology, and if you think about different features that a human has, there could be so many things that one could differentiate based on the genetic level and also the physiological level or phenotypic level. So there is a lot of variety of data that we need to make sense of. So if how height is related to weight, if a person is more taller, will his weight will be high or not? So these type of patterns you could see from from data. And then there is different types of genders, like if you include male, if you include female, then all of this data will have a lot of variety. And for us to understand and make sense of it, we have to connect these different types of data with each other. So a male in this type of population will have this type of things. So all of this data connects with each other. So we need to understand, and that connection is what we are looking at. So when we are looking at big data, we are finding two things. One is pattern, second is connection. So these are the things that our data scientists look into and are really get excited, because these leads to discovery. So I told you about variety, I told you about volume, then there is value. Because there's a lot of data noise also. So because of different types of technologies has pros and cons. So all of these technologies where there is high throughput data is, kind of generated, we have to use different types of algorithms to make sure that the data that is coming out of the system or the machine is making sense, because there are different types of artifacts, there are different types of noises, technical errors, and there are a lot of things that actually happens in really when you do this practically, you would know, because we'll also have a practical session, which is going to be really exciting after learning all of this, because then you will be seeing that what this data actually is and how it looks, and what are the different ways to analyze this, what are the different algorithms that actually cuts this into make it more meaningful. So these are different challenges that comes into this, and that's where our company uh, started from. So Pine Biotech or Omics Logic is a bioinformatics and data science research training company, but it started from a technical spin-off from a university, which is University of Haifa, at the Tauber Bioinformatics Research Center. So Dr. Fred Alfred Tauber, who is a philanthropist, and also is a expert. He's an MBBS doctor, and he has different types of. He's a very renowned researcher. His record of research citation places him in the top 2.5 of active investigators in the world, and has been doing a lot of different types of research project centers and development of different types of bigger opportunities. So he funded this Topper Research Center, and the idea was to simplify this complex research, because when I talk about doing one kind of big data research, then it is not just that you will use one tool and then you will get the data and you will have everything simplified. It is not. So it is a pipeline of algorithms. So data runs from one tool, then it goes to second, then it goes to third, fourth, fifth, sixth. So it is refined, it is getting refined and refined, and you're trying to do then different types of things. Some type of algorithms will give you some other type of analysis, some type of algorithm will give you some other type of analysis. So it's an algorithmic approach, and in this algorithmic approach there are workflows. So how does this workflow works? It is like raw data is coming, and then you are getting to different types of structured data, and then you're getting into biology, then you're looking at like how this is like making sense in biology, what is happening. Then you go into discovery and understand that, oh, this is a new thing, this is what I am trying to discover, and this is what I have learned, that this data speaks about this type of things. So this is how the new data driven world works. So these guys actually were motivated to make it simple for the clinicians, for make it simple and make democratize bioinformatics. Democratizing bioinformatics and data science means that making it available for everyone. How can we do that? How did they did that? So they did that with the help of Dr. Leonard Brodsky. He's a mathematician and a brilliant scientist, who wrote these different types of algorithms and made it possible by providing a graphical user interface that is like a point and click system through an AI based approach. And now it's been run by very capable scientist, Dr. Julia Panau, who is a very close collaborator, and she's helping Pine Biotech and Omics Logic with research projects and also education, and with her suggestions, and including myself. So I'm heading the US and India company right now, and collaborating with several universities as well as research centers in the US as well as in Israel and in India, by utilizing all of this resources that I have with the help of this great team here. Now what they have done is that they have kind of prepared this this is one of the things that they have done. So as I was telling you that many people are not comfortable with coding, so they developed a way so that every biologist or clinician can do big data analysis with or without coding and get comfortable with it and get like really fine with all of this. And then, I mean, even in my experience, I mean, I used to get scared with many of these things which I was not comfortable with. Recently, for example, finance. So as I'm running a US company, I need to know all the taxation and all the finance and whatever is happening. And I was never comfortable with it. But I thought that this is the time to learn, and I really started enjoying it once I got comfortable. But initial phase was really challenging. So I think that's the barrier that you all have to jump. That you have to be comfortable and get some rough hours there and have patience, and then I think you will be able to take those big jumps and do whatever you are like kind of hoping to get out of your next 5 years or 10 years or 20 years. But to help you, this is just the best way to start this, because it has a simplified way, makes you to learn the logic of analysis rather than going into the complexity of building a thing like this. Because many times when we start research, we start from looking into the literature review, then we go into Google and find out what is there. It's a very long cycle. For a one person to do a PhD, it takes 5 years, and this technology is rapidly evolving. So my suggestion is to start as early as possible. Many of our students are high school graduates, and these high school students are now like they started in their high school and they end up doing a master's or PhD in Harvard or MIT or like some of these big schools. And I'm really proud to have these students throughout. Like when we started in 2018, and now we can see that these students are making a mark. So I think it is kind of important to start early in this new modern way of evolving with the education and the technology. And this has been possible only without a great team that I have here with me. So all of this credit goes to them as much as it goes to anyone else. So we have been working really with several of these research institutes, understanding what are their needs in terms of big data analysis, and then converting that in experience into educational way, so that students can learn and be really ready to fit in, and where these major gaps are there in bioinformatics and data science education. So we collaborated with a lot of universities and started several programs, and had an opportunity, obviously, to work in Amity University, Rajasthan. And we are also planning several new things that would help the students in the coming year. But it has been really good experience while collaborating with some of the prestigious institutions such as All India Institute of Medical Sciences, IIT Jodhpur. We are starting research and training and PG diploma program, and different regional universities as well. So as I was saying before, that there are two pathways, and the coding pathway is sometimes you would see a lot of emphasis put on that, but you can start from no code and then you can gradually go into the coding pathway. But many of you are more interested in like learning and are quick learners for coding. That is also possible. That's what we have been able to develop along with the two different teams. So working with the teams in US, working with the teams in Israel, working with the teams in India, and all the researchers and collaborators from UCSF, from Harvard, from Louisiana Research, Louisiana State University and Biomedical Research. So all of this led to development of these type of platforms, where all of this different features are included. Like this is a way to provide bioinformatics modern education, and we are trying to build a platform that includes all of these components in one place, so that the user or the student does not get confused and does not get lost in this process. So all of this, we structured it into different types of curriculums that we are piloting and that we are running in different universities and training centers. Now, this led to a lot of recognition as well in terms of what we were able to achieve. So over the last years, we have collaborated with over hundreds of organizations and researchers solving complex biological problems and developing cutting edge solution and services that could be utilized, that has been utilized by universities. And we got a few awards, although we barely get time to do all of this. So most of our focus is to provide and do this continuous learning and be a part of this cutting edge research. So the technology is being utilized by hundreds of students and faculty and researchers from a partner network of universities and academic institutions that is leading to translational bioinformatics practices and education, as well as with industry. So collaboration with bioinformatics and data science industry, as I was telling you, that the for jobs, for internships, for industry expert sessions, for data driven research, and then for career guidance and placements. We also have a partnership with another company that provides a very good insights into how can you best prepare your CV, how can you best prepare your cover letter, how can you apply for masters and PhD programs. So we also integrate their offerings in our program so that a student can not only learn but also go to the next step right after doing this. So that was about us and how we started and what we were able to achieve in this short period of time and where we are heading. So one of the reasons to do this is obviously to contribute first of all, and utilize this experience that the center is having with their platform and technology, that they're constantly evolving. So they are working with Roswell Cancer Center in the US. They're working with Path, an organization where there will be vaccine testing algorithms and pipelines that will be utilized by FDA for the testing. So all those big projects is what Tauber Research Center is being able to work, and we are fortunate to be able to associated with them and learn all of this as well as implement and do some of those projects ourselves in the future. So now coming back to where we were, I mean, about the why we should do this. So I had addressed a few things, but I have not addressed well that why as a student you should be looking at and trying these things and learn these things. So we learned about that bioinformatics is the intersection of informatics, or the science of processing data for storage and retrieval, and biology, and then study of living organisms. So what possibilities the modern science has opened, and why we are adding these innovative curriculums to supplement basic science and degree programs. To answer that, you have to imagine a world where we can decode the genetic information stored within every living organism, from the tiniest microbe to the largest mammal. A world where we can unravel the mysteries of diseases, predict their onsets, that when predict where this disease will be, and then devise customized treatment. Like, that would be a really nice thing to do to the humanity, and everyone will be really less burdened with all these diseases. Some of these diseases are really such a big burden for families that they cannot go beyond and sustain. So bioinformatics is the key that unlocks these possibilities. It's where computational techniques meet biology, where data analysis leads to groundbreaking discoveries, and where innovation has the potential to revolutionize healthcare, agriculture, and beyond. So how it is possible? So just imagine, like, when you're doing something, for example, how you can make a meaningful impact. So in this field, every line of code, every algorithm, every analysis you conduct can contribute to saving lives, and can contribute to improving health, and can contribute to enhancing your understanding about life itself. So your work could lead to discovery of new drug targets, the development of personalized medicines, and the identification of biomarkers that signals the presence of a disease. These are very common things that many of these researchers are doing. But not only these, you can be a part of global challenges to solve these global challenges that the humanity is facing. The world faces pressing challenges in healthcare, environmental sustainability, and agriculture. So bioinformatics offers solutions to address these challenges, from understanding the genetic basis of the disease to designing, for example, resilient crops that can withstand climate change. And by joining industry, you can contribute directly by solving these global issues. One of the other advantage of being this field such an interdisciplinary, so there are like pros and cons. I told you cons that you have to learn a lot of things, new things, but there are also pros that we collaborate a lot, that we know that we don't know everything ourselves, so it's best to collaborate and use our best of our abilities. Similarly, that's how we have collaborated with this workshop, and this workshop is a collaborative possibilities what we can do together. Similarly, in sciences and in research, bioinformatics is not confined to one discipline. It bridges biology, computer science, mathematics, statistics. So by engaging in this industry, you will collaborate with experts from various fields, and that will broaden your horizons and foster a very rich learning experience. Another aspect is innovation. So we discussed a lot about innovation. So the bioinformatics landscape is ever evolving with advancements in technologies like next generation sequencing that we are going to discuss, and high throughput data generation in the next few sessions. There is always something new to explore. And that's what this big data has given us also as an opportunity, because earlier it was very difficult, like whatever I'm doing right now, it was very, very difficult 20 years ago, because there was no big data repositories as rich as what is right now. And what happens in big data is that people work on specific set of experiments, they do it and they prove their hypothesis and they publish and they deposit their data. But that big data is about the entire organism. Think about that. And entire organism, and you are just looking at, for example, what happened, what are the effect after I got COVID-19 infection. But that person might have other comorbidities as well. So in that case, you would also learn about different types of other pathways that are that are affected. So there is always this possibility of discovering something new by scratching the big data, by finding meaningful information from the big data. And know, like, you should know beforehand what you're doing. You can't just go there and big data and think that things will happen on your own, or you'll suddenly find something. It does not work that way. So what actually works is that you have to be very specific, and you then use your expertise, develop these type of collaborations, and then you reach to your goal. But you have to be very sure about what you want to find, because that data has a lot of information that might take you you get distracted and you'll not ever reach to the conclusion. So now the different aspect, we discussed about all of this a lot, and now about data science and not programming actually. So with the outbreak of data science revolution, a war between R enthusiasts and Python fanatics has emerged. So as a result, Python and R have been compared and contrasted 1,000 times with a detailed listing of respective advantages and weaknesses. So this warfare led to misconception that as a data science learner or enthusiastic, you should relentlessly focus on either mastering R or Python. It's a bad advice, because the actual key to becoming a data science professional is in understanding the underlying data science concepts and work towards expanding your maybe toolbox as much as you can learn. So it is not about learning only specific type of programming language that would make you expert. It is never about that tool. It is about also learning the logic behind the tool. So that's why we call ourselves omics logic, because we focus on the logic, and then you will be able to use any tool. But yeah, I mean, R and Python are something that we cover in our coursework as well. because they have a lot of, like, they help in, there is constantly new libraries and new type of uh, features that people are like working on and developing, and a lot of open source stuff is available. In addition to that, you can leverage them to do a lot of visualization or data wrangling that actually helps in publications, that also helps in grants and all. So that is also useful in that context. So in terms of, understanding also the language in terms of job opportunities, because when we are learning job languages, we are looking for these skill sets that you will see in a job profile. So we often see those things, and we kind of, this data is coming and showing that what kind of jobs are out there in terms of coding. But when we talk about you being a biologist and people who are interested in biology, it also helps in that. So the learning not only helps in getting better opportunities, but also helps in reducing the experimental cost for the research and products. How so? So as NGS is making its mark in the field of diagnostics and testing, just imagine, like, all of us are going through some sort of diagnostics or testing when we are getting sick, or regular testing. This is a common practice, and it's going to grow further, because by doing the diagnostics and testing, we are generating data about ourselves, our body. And companies, and NGS is making a mark. So NGS will be one of the biggest benchmarks in diagnostics and testing industry. So companies are expected to hire experts who can work efficiently and save large amount of resources by not failing to do the experiments, and achieve projects in very affordable cost. Just like ISRO did with the Chandrayaan 3. So those are the things possible with the knowledge, and with using and to be knowing what technology is there. So these are the things that we are doing in an omics project education setting, is to understand, to be or not to be. Should I become an expert or a generalist? My future academic career, courses, lectures, training, is it enough? Projects, how that is going to help in building a great profile and a CV, and cover letters and things like that, that would help us go to a better organization, a bigger organization, which is making a mark in their world, in in revolutionizing these technologies, like Google and other institutions, where we can get an opportunity with what we have learned, and then filling this gap, and then by doing experience building opportunities through internships, because it is kind of important for you to know what industry is doing, and by learning by doing. So that is something that many of industries and these different types of organizations are looking into. So different types of bioinformatics positions are available. So we have a list which I show you in the job support. But what Omics Logic is doing is that it's providing a soft landing into the field of bioinformatics and a complete pathway from learning to application. So we have developed resources, coursework, and there is a pathway. You go through this path by basic training, and then you learn about these basic concepts. And these are just focused on a practical application. So it's not theory, but it's all practical based. So you kind of learn about these different types of uh, concepts in a way that anyone can learn. So basic training helps people without any background to go into this field. Then advanced training, you choose what is your interest, and then specialization is where all the interesting things started. In all the interesting things starts to happen in terms of doing research in an area that interests you the most, and then recognition and how to achieve that, and then after that you get recognition for your work. So what we are doing is providing these type of research internship tracks as well, so for students who can develop independent research project proposals focusing on industry or academic or industry tracks. And we have a lot of collaborators, like big names in here, like Carnegie Mellon, LSU Health, Georgetown University, Roosevelt Park, NMIMR in Ghana. And we kind of connect these industry skills as students complete, their journey. We track those and we complete those complete profiles and develop a project portfolio for the student who are going through this journey, so that company can find these unique skills at an affordable rates and finding their work and tracking their skill mastery, so that it's all feasible. And this is how industry would kind of do this, in terms of matching the skills of what is required and what you have gained. So also, which leads to different types of job opportunities which are there in the US. There's a lot of opportunities, and there is a lot of like there's a lot of in terms of salary and all, there's a lot of difference. But India is a growing market now, and in India also we are seeing a huge paradigm shift in terms of job opportunities and different types of technology based opportunities and different types of things that are required. So you would say that in different types of job opportunities, these type of skill sets are needed. So in terms of degree, in terms of communication skill sets, in terms of learning about different cloud technologies, database experience, tools and packages, to make sure that we are heading towards that based on the student, based on their interest and how it could be customized. So learning and adding to your profile is going to be important in the future, to have a good CV and also to have it presented well in different type of organizations where you are going to go next. So connecting the information with your skill sets is something that Domix Logic is trying to do a lot. And communication and editing and developing tutorials for this same, we have a team who is responsible and working to make sure that whatever new knowledge is coming, we transfer that in a way that is more meaningful for the student. And similarly, we kind of make sure that you go to the next level by doing a practice, by learning, by doing yourself and working on projects and publishing these results, in terms of getting research experience, so that that could be counted as well. So this all resume building that reflects your background, that shows how to find relevant jobs, and all of that, I think, is something that we wish to cover. And we are like really excited to be able to do this in the coming days, where we are having these discussions. I'll come to that. But bioinformatics in industry, I would say, is helping to build domain specific expertise in addition to background in data science. So one example is technologies used in genome assembly, which is the process of deciphering sequences of genetic material within an organism. So these applications are from these different types of companies like Nanopore, Illumina, PacBio, and as a data scientist working in a field outside of biology, you may not be familiar with them. These are big companies who are doing a lot of research work, and where a lot of opportunities are also there. Another example of domain specific expertise is writing code. So that is, as you look at the transcriptome or readout of all the RNA that is present in a cell, you need to know how to perform tasks as like, aligning sequences of genes or annotating proteins. These tasks require not only, writing scripts, but also usually you are trying to, do use whatever is available. So many of these scripts are and tools, how to use them, how to use the libraries, and do the analysis in terms of market size and jobs and salaries. So I'm showing here two types of average salaries. One in Indian market that we recently did a survey. One in the US market. So in the US market, these are taken from salary.com, and bioinformatics scientists average salary is about almost $100,000, which is equivalent to maybe 82 lakh a year. And then there are different types of job opportunities like research scientist, where the base salary is $83,000 and plus, and then the biostatisticians, and then microbiologists, and then bioinformaticians and zoologists and wildlife biologists who are working and using data, molecular biologists who are using data. So all of this is like now considered into the same bucket, that if you know data science skills and if you know bioinformatics skills, your salary will be much higher than these different types of only like if you know only one skill. So data analysis and data science skill sets are going to be a huge factor in getting to good salaries versus normal salaries or below par salaries. In India also, for data science, there's a lot of goods packages and salaries that companies are offering. And as well as in research institutes, there are a lot of positions that have come out for such opportunities. So this is what I think the idea is, to be able to give you those important skill sets that will be relevant for your job search, for your next educational search, or whatever that you think is more that you want to look into. So Omics Logic has these different types of ways of collaborations, and also different types of trainings and research platforms that allows students to go through this experience in a fairly short time. So what we have been able to achieve by doing a lot of automation, a lot of AI, and a lot of graphics, and by putting a lot of effort into this designing curriculums and all, is to be able to do this in 3 to 6 months, which is what we've been able to achieve. And I'm really happy that with Amity University, Rajasthan, and with the guidance of Professor Sharma and Professor Neelam and Dr. Ravi and other the respected VC, we are kind of planning to take this forward and develop a joint certification program in biomedical data science in Python, which we are working towards, and it's coming soon, and we'll be sharing more about it once it is once it is out from approvals and all, and ready for students to take. So we are focusing on biomedical data science in Python, where a lot of Python related collab notebooks and playing around with it, and like learning AI and the use of artificial intelligence and things like ChatGPT, and learning how to use this large models and get to the best of data is something that is really a very new thing to the world. And we have been able to not only do it, but also integrate it in our education. So we about that, and also we have these upcoming semester coming up with LSU Biomed. So it's a group of 29 universities where these different fall programs will be available by Omics Logic and LSU, Louisiana State University Biomed. It's a network. So introduction to multi-omics curriculum and genomics curriculum, metagenomics curriculum, and the Python curriculum will be available for students who are from this network of universities. Similarly, National Association of Idea Principal Investigators, where we have collaborations in 23 states of the US and Puerto Rico, and the PIs from there and the students from this National Association NEPI network will be able to join these different training programs that will encompass for 3 months, will include live sessions and also recorded and course material to finish and at their own pace, and complete it along with their degree programs that they're enrolled in, or the scientific work that they are doing. So I think for students, we also have the research fellowship program. The research fellowship program is a very special program who is actually more focused on finding research through this different types of learnings. So I would like to give, as this is my second last slide, and I'm going to conclude this session and hand over to my team to go for more interactive and practical sessions at 2, and before that, more interactions and completing some of the things and resources, providing you with the resources, free resources, talking about the WhatsApp group and different things. So that's the exciting part. I think the lecture parts are getting over. But I wanted to end up by giving you an example. I know that there is a lot of students in food technology, microbiology. So I wanted to talk about the application of next generation sequencing in food biotechnology because it's vast and continuously evolving. So in food technology, for example, I'm here. What we are doing is also working with students for a period of 2 to 3 months and trying to come up with a quick project that could be worked on further and developed into a publication or developed into a report. So I think in food, for example, food technology and food biotechnology, um, this type of analysis, next generation sequencing, is used to identify and quantify pathogens in food products, helping to prevent foodborne illness. And then there is a technology called microbiome profiling, which is understanding the microbial communities present in different food products, which can influence food safety and quality, as well as authenticity and adulteration. And then there is species identification, like organisms present in food, to help different types of understanding their value, nutritional values. So, and then there is geographical origin verification. That sort of things can be also done through food technology and food next generation sequencing technology, identifying the genomic markers. Then there is nutritional genomics. So nutritional genomics is the study of interaction between genome of an individual and their diet. And there's a huge market, I must tell you, about the microbiome and nutrigenomics, because it's also used in sports, because a lot of people who are into that are looking for specific types of diet that could enhance or at least increase their abilities. So this personalized diet is something which has a huge market, because you could think about, people are trying to lose weight, right? This is a huge market. Many companies are working on it. Many products are there. Thousands and thousands of products are there. So microbiome based analysis can tell you exactly the effect of food, exactly your food microbiome, gut microbiome. Just imagine the application of this. If people can, if you can give people based on the genetic information, that would be the most reliable way of giving them recommendation with all that data. So many companies are working in that aspect as well. So I think huge opportunities in the future in terms of where the industry would be heading and where the job opportunities will be. So another aspect is genome assisted breeding. So using NGS to identify genetic markers associated with desirable traits, which can assist with crop breeding programs. Then GM foods, like developing genetically modified foods with improved nutritional profiles and resistance to pests and diseases. And then fermentation of food processing, then strain improvement, and then soil microbiome. And it's just like a lot of things are there, which I can keep on going and going about it. So in terms of also global warming and all of that, and the need of food with the growing population, I think this is going to play a huge role. And the companies who are going into the food, a lot of companies and startups are there, people from abroad coming into India and doing agriculture, and they are willing to introduce all type of technologies that could enhance their the efficiency. So I think what we do is also, in terms of other than curriculums, we are doing these type of structured short training programs, and then the whole world is now heading towards it. So I just wanted to end this session by concluding that AI is changing the pharmaceutical industry, and even generative AI, like after AI, generative AI can turn every biologist into a computer scientist. I tell you that biology is important, but with automation and all, data is more important, because in the end, what we do in biology is generate data, because all the experiments that we've been doing from single cells, from single organism or single protein experiments to now high throughput experiments, it's always about data generation. Just to give you a small case study. So a team of researchers at the MIT, they used AI to discover an antibiotic capable of killing E. coli, which hospitalized thousands of people a year. So you know how important E. coli is, and if some antibiotic is killing it, and then it's something that is having a problem in our food, in our digestion, as well as antibiotic resistant strains of another common bacterial infection. So this is Acinetobacter baumannii, and they discovered that this helicin, so this is also called helicin or HAL 9000. So this discovery of helicin paints a picture of just how rapid AI-assisted drug discovery can be. So scientists trained their AI models by introducing it to, in this case, they trained it with approximately 2,500 molecules. 1,700 out of that were FDA approved and 800 were natural products. So FDA approved means that there will be no issue with the side effects and all, and natural products will not have side effects. So as the researchers trained the model to understand the molecule would kill E. coli, the team ran 6,000 compounds through the system, including existing drugs, failed drugs, natural products, and a variety of other compounds. The system found helicin is in a fraction of time compared to the traditional methods would take. So so this the faculty at the university, there's a faculty in this university who teaches how AI is changing the pharmaceutical industry, and told about this specific type of breakthrough, that that discovery is important because of the antimicrobial resistant, and also incorporation of things like superbug, or things which could be has a devastating effect on our systems, on our healthcare. So there are a lot of deaths that actually happen. So for with at least, like, I think if the number is correct, 700,000 deaths every year contribute to drug resistant diseases, and this is projected to grow a lot. And in this case, I think AI is going to play a critical role, and this and the history, and especially with the large language models and image generators like OpenAI, ChatGPT, Google Bard, Midjourney, introduced through the concept of AI. So I think it's going to be very important in understanding and doing this fairly quickly and at a very, very large scale. Like, not thousands of molecules you taste, not lakhs, not crores, but more than that. Like there was a recent study by Regeneron. They kind of screened 16 billion compounds. Just imagine, which would take maybe 100,000 years before, now in a month's time. Also, quantum computing is making a huge leap in this, and the computational time is going to change. Things will be different, and we are looking forward to all of that. But to stay competent, I think it's important to not ignore this, but to accept it, and then try to figure out the best way to kind of implement it for the better of humanity, and think about the consequences before doing anything. So I would also say that not doing it because of the money it pays also, but also you have a responsibility towards the society, towards the earth, and towards the entire changing paradigm. So I think this young generation is going to be critical for that, and we are here also to help you out with the learning and your putting your starting steps into this journey. So with that, I'd like to conclude my session. I hope that whatever I have said has some inputs and impact in the students who have joined and learned today. And I'll be happy to connect with you later also, as we are collaborators with MIT University, Rajasthan, and we keep on having different types of events and also planning to start this credit-based course. So we look forward to meet with you, and please feel free to write to me or connect me on LinkedIn, and you can reach out to our team also. So thank you everyone for listening, and I'll hand this over session to Ms. Srigori, or and then you can also put your questions and things on the chat that we can address likewise. Thank you all.
Okay, thank you so much, Dr. Mohit, for that wonderful session. So before we dive into the practical hands-on session, we will first start with a small activity, right? And then we will try to understand, so far we learned what is the growing emergence of the field of data science and what are the various learning resources available. But most often students struggle with how to leverage these learning resources to get started in. So that is what I'll be covering in my presentation. So before that, I'd like to encourage the participants to first visit the website, that is Omics Logic Learn portal. Let me share the link in the chat box. Okay, so this is the online learning platform where you have access to all of the learning resources on data science and bioinformatics. So to begin with, for those of you who have not created an account on the Omics Logic Learn portal yet, I'll just briefly walk you through on how to get started, right? So I hope all the participants are on their laptop. Can you confirm that in the chat box? Please put a yes in the chat box so that I can get a fair idea about that.
Excuse me, myself, Dr. Ravi Ranjan. Probably chat box is disabled. So let me ask IT person.
Okay, thank you so much. All right.
IT MIT University. Can you hear me? Just wait for a moment.
Yes, sir.
A few participants is saying that chat box is not enabled. at their end.
Sir, the participants can also post their question in Q&A.
Okay. So question and answer, right? Okay, so dear participant, you can post your text in question and answer section, right? So
Now, sir, chat is also enabled, sir.
okay, and now chat box is also enabled. So thank you, IT MIT University.
Thank you so much.
And to you, Shri Krishna.
Thank you so much. So I'll just pause for a couple of seconds so that the participants can log into your laptop and get started. So this will be a small activity the participants can get started. And in the next session, we'll be having an incredible practical session by one of our excellent training instructors at Omics Logic. So sit back and enjoy, and ready to learn a lot of new concepts. So let me know in the chat. I'm just waiting for your responses. If you're logged in on your laptop, let me know with a yes. I'll just paste the link to the Learn New ID in the chat box. So this is the link for signing up on the Omics Logic Learn portal. So in the next session, the mentor will be covering a lot about the next generation sequencing. And while along the way, if you want to refer to some of the topics, then you have the opportunity to go through some of our open courses on the portal. So to know how to navigate the portal and to get started, I'll just walk you through a brief demo on how to do it. So first of all, you need to create an account on the Omics Logic Learn portal to get started. So to create an account, you can click on this button that says create account. And here you will be asked to enter your name, email ID, password, and then few other details about your education background So once you submit it, your account will be created. Or let's say you can also directly log in through any of your existing social media handles. That could be your Google account, Facebook account, Apple account, GitHub account, or even Twitter. So this is how you can sign up on the Omix Project Learn portal, right? And along the way, if you're having any technical issues while signing up, you can let us know in the chat as well. Our technical team is here as well, so that they will be able to look into any specific issues that the students have. So let us make it more interactive. Please don't feel shy or anything. Be sure to be interactive and make the most out of the opportunity that is provided to you by your institution. So once you have created an account on the portal, you can put a yes in the chat box. And if you already have an account on the portal, you can put a done in the chat. Okay, thank you so much, Dr. Ravi. We have Ritika has also signed up. Okay, all right. So I'll just proceed along so that if at all you are having any queries, please feel free to let me know in the chat and I will be able to guide you on how, right? So since I already have an account on the portal, I'm directly proceeding with login using my existing login credentials. So once you sign in, the very first interface that you are seeing is the course page, right? So here you have a library of collection of online courses, example projects that are based on bioinformatics, genomics, metagenomics, transcriptomics, and so on. And if you head over to the, when you scroll down below, you will see a search bar that says looking for a specific course, right? So if you are completely new to the field of bioinformatics, and when the mentor is referring to some topics that you might not be familiar with and want to refresh your course on that, then you can head over to the courses tab and then directly enter the keyword Introduction to Bioinformatics, which is an open course that anyone can enroll, complete the lessons, and get the certificate, right? And then you also have another introductory course on bytes and molecules, which will introduce you to various specific subdisciplines within the bioinformatics, such as precision medicine, phase omics, infectious diseases, how these bioinformatics techniques are used. You will get an overview about all of the bytes. Now, if you're someone who is interested in coding, right? Let's say you are interested in Python, then you have the open course Python course 1, getting started with bioinformatics, which will introduce students from a diverse range of background to the field of how is Python used in bioinformatics, and then let's say you are someone who is interested in learning R coding, then you have the open course R coding course 1, getting started with. So this is how you can navigate through the portal to refer to the open courses that are available and which the mentor will be making use of during her practice. And then if you are looking for a specific subdiscipline within bioinformatics, such as genomics, then you can enter the keyword genomics. And then you will see the associated course, which is course 3 genomics. And then you will also come across several case studies where genomic techniques are being used in various settings. How bioinformatic techniques are used, tuberculosis treatment, how it is used for gradient calling analysis, oncology research. So you can have a look at all of that, and I'll take just one more example of transcriptomics. So transcriptomics, which is study of gene expressions, then you can go to how gene expression studies are used to understand post-pathogen and liver cancer. How you'll be able to navigate the courses, right? So now that is about the courses. So one of the very important things that you have to do once you sign up is to set up your profile so that we know who you are and we can provide you with the personalized. So how do you set up your profile? You can click on the top blue bar that is welcome back with your email ID. And then when you click on that, it will display the profile setting. And here you will see the profile button, and here make sure that you have the profile image, right? And add a brief bio about yourself, about your educational background, if you're from a biotechnology background or microbiology, food technology, mention that, and what are your research interests, right? And then also feel free to link your social media handles so that you can connect with like-minded individuals who are part of the OMIC project. And you will also see various sections within your profile, which is the activity section. So this is one of the crucial sections where the mentor and the community manager, they will be able to track your learning progress. So you could see the live activities of the participants here. So when we see that they are stuck with some particular concepts, they are not able to progress ahead, we reach out to them and provide them with assistance. And under the courses tab, it will display the various courses that you have currently enrolled, as well as the progress that you have made. And then under the certificates tab, once you complete all of the required quizzes, activities, assignments that are there in the particular coursework, you'll be able to access and download the certificate of the certificates, right? And one of the very exciting sections that most of the students will be interested to navigate is the projects tab, which is there on the OMIC's Logic Learn portal. So I'll be covering a lot of these case studies of students, how they have leveraged the training resources that are available to them to transition into the field of data science. And there are so many incredible ways that the students have done that, and I'll cover that in my presentation in this while. But if the students are interested to check it, I will just share the link in the chat box. So here's the link in the chat box for the project section. And here you will see projects that have been completed by students in various topics, such as immunology, cancer, food technology, machine learning, infectious diseases, neuroscience, and so on. And all of these student projects have been structured in a way a research publication is done, so that students who are interested to get an idea about how to carry out research, they get it. So I just quickly open one of the student project and I'll show you. So as you can see, the students, there's a brief recording by the presentation by the student who will explain the topic, the research question that they have developed, and the various analysis techniques they have used. They have uploaded the data set, so the students can download it and see what kind of analysis they have performed, brief the literature review that they have carried out, an introduction about their project, methods, that methodology that they have used to carry out their research project, which includes several visualizations from the pipeline and results. So this is just what I wanted to briefly go over in my topic regarding OMIC's Logic Learn portal, right? Now I'll just quickly head over to my presentation and help you understand how the students have leveraged these resources, right? So as previously covered in the presentation, this is an online learning community, and there are so many users. There are over 24,000 users from over 30 plus countries, from over 300 programs. And I'll just give you a brief roadmap about how the students progress in their learning journey who are completely new to the field of bioinformatics. So once they enroll for the training program, they are required to watch the associated lecture videos. The mentor will be providing them with associated lessons that they are required to go through, which will supplement their learning experience while they're watching the lectures. And then there are also scientific articles that the students can go through to get a further understanding about the field. And most often than not, while learning online, students do get stuck in a particular topic, right? And it might be, let's say you are stuck in executing code on a problem. So we have provided the online assistance to help the students get started with their queries and provide them with assistance. So here is a screenshot from some of the queries that have been reaching out to us from one of our recent collaborations, with Louisiana Biomedical Research Network. So this includes researchers, professors, and students who reach out to us with their queries, and we assist them through the online tests. And we also regularly check for the students' feedback so that we can optimize the courses, so that we meet the learning experience of the students. So if they find a particular topic complex, then we make sure that we try to simplify the concept so that it reaches the students and make their learning experience better. So this is one of the recent feedbacks from the training program that I was just talking about, right? And then we also check for the difficulty of the courses and help optimize the curriculum. So we have had the opportunity to work with several of student communities from different universities, and the students who are able to perform very well, we are able to provide them with a report, a certificate of excellence as well. And as you can see, these are some of the top performers from a recent training program, Leva University, Louisiana University, Stella Maris College, Asian University, and so on, right? And then how, and on my next slide, I'll just walk you through on how these students have leveraged these learning resources in their, to upskill themselves. So I'll just start with a very basic program, which is the Introduction to Bioinformatics, which most students might be interested to go over, right? So Introduction to Bioinformatics is the field that students most get overwhelmed with because of the complex terms and topics that we exist. And that is why we have simplified the course with particular meticulously designed curriculum to make sure that the language is easy to understand and students are able to easily go ahead. So along the testimonials, we will come across all of the students from computer science background, mathematics, and so on, so that they are able to get started in the field. So here's one of the testimonials from the genomics stream. And one of the student has been from MIT University itself. So she was from a biotechnology background, and she wanted to carry out a infectious disease project using genomic data analysis. So this is the training that she has opted for. And from understanding how to identify the genomic data sets and what are the various techniques that we have to use to analyze them, all of that will be covered in the training. And these training programs will also be especially a very great refresher for the concepts. If you're someone who is interested in transcriptomics, then there is a very refresher session in the beginning, which will help you memorize, recall the various topics that are covered in morphology. And if you're someone from computer science background, then you will be able to grasp the knowledge very quickly because. And we also have the metagenomics students sharing their testimonials as well. We'll be covering a lot of these case studies in the upcoming space. And cheminformatics is also one of the popular topics of participants from various training programs who are interested in structural biology and computational chemistry, right? And as Dr. Mohit was mentioning in his previous slide, the research fellowship program is one of the most popular program that is there amongst the students. And here in my presentation, I'll just walk you through what are the various specialization tracks that the students have opted for during their training program. So initially they complete their training in a specific field like genomics, metagenomics, transcriptomics, as I was mentioning in my previous slide. And then they choose to specifically specialize in a research track. And these include precision medicine, population health, biotechnology and agriculture, and space and astrobiology. So we'll just go over some of these case examples. So here is one of the training collaboration with systems medicine graduate program. And here the students went over the Python coursework to develop machine learning models to address various research questions in diverse disciplines. Right. So for example, Rohan was one of the students who was interested in developing a machine learning model to differentiate between asymptomatic and symptomatic COVID-19 patients using transcriptomics data. And Rima Ahmad, she had also done a very wonderful project She developed a machine learning model to predict whether an individual has diabetes or. And Jordan was another student who developed a machine learning model to for post-chemotherapy for neurodegeneration. And Michelle, who wanted to understand the post-traumatic stress disorders, the unique features that are pertinent to those individuals. Like that, you will come across several of these example projects in the link that I've provided to you in the chat. So please feel free to go over it and explore their projects. Now, students have also went on to exhibit their research projects in Research Symposium, and we annually conduct the Omics Research Symposium, which brings together students and researchers and faculty from all around the world, and they present their project and win exciting cash prizes for them. So here is one of our research fellow. She was a PhD student, and she wanted to transition to the field of data science, and she opted for the Python coursework and developed machine learning model for drug prediction. So this project is available on the portal as well. And then there are also some students who are interested to complete a research publication. So here is one example of Ricky Vajra. She was very passionate about research work that she was doing in immunology. And initially, she took the transcriptomics training. She understood how gene expression studies are done. And then she selected her data set from immunology. And then she went on to work on a multiple sclerosis research project. And then there are also students who are interested in carrying out a cancer project, such as Sine Doraner. So she was from a molecular biology background from an undergraduate student from Turkey. And she went on to carry out a research project in cancer and published a project as well in Columbia Undergraduate Science Journal. And then there are also, and one unique aspect is that these programs are not just restricted to undergraduate and graduate. There are several students coming from the high school as well. So this is one example of Andre Modena Sosa that Dr. Mohit was mentioning in his slide as well. So initially he learned about the transcriptomics, the gene expression studies, and then he went on to complete a research project and present it at an international science competition for which he won the Potemkin. And recently he is enrolled in a wonderful program at Harvard University in chemical biology. And this is one of the case studies that might be relatable to all of you. They were students from MIT University, from biotechnology background, and they also were looking to transition into data science field, and they worked on developing machine learning models, predictive models, and they also went on to publish, present their project at an international IIT conference for which they won the certificates. And presently they are working towards publishing their And there are also students who enrolled for the research fellowship program to complete a research project, and they showcase their research project as a part of their higher university college application. College applications can be very competitive, and research project can be a very stepping stone for students to get started. So in my next slide, I will just be taking the example of Dilara Daikin, who completed a research project on space omics. And you'll find several of these student club videos on our YouTube channel as well, where she is talking about her passion for astrobiology. She went on to work with metagenomic data sets to understand humans in space. And this is another example of Subhajit Barua, who is a very active person who communicates about his passion for astrobiology and space on various communication channels. And presently, he is enrolled in a PhD program at the University of Pittsburgh. And then if you're someone who's interested in analyzing infectious diseases using bioinformatics techniques, we have also the several researchers who are interested to do that. And here is one example from a team of researchers from Neguchi Memorial Institute at University of Ghana, who went on to complete a infectious disease research project using various computational-based techniques. And then there are also students who come to complete a research project with a passion for business as well. And in such cases, here is an example of a team of industrial researchers from Nigeria who enrolled in the metagenomic data analysis training, and they participated in the hackathon, and they developed a microbiome test kit to improve the chances of women in Nigeria to carry their babies. which is an excellent contribution to the scientific community. And one of the unique aspects is that these projects are not just completed by researchers and industrial researchers. There are also undergraduate students and postgraduate students, such as a team of students from VIT did. They came together to develop a test kit for early detection of Alzheimer's disease. So all of these projects are available on the Women's Project Learn portal. So I would highly encourage you to walk through all of the projects that students have done, what are the various research questions that they have come up with, and how they have decided to showcase their projects. And a lot of these research opportunities and student outcomes, they are presented in our LinkedIn and our social media handles as well, where students actively share their progress updates and the certifications and how they have presented their projects in various scientific opportunities. So that was all about that I wanted to share with you in my presentation. And for now, to get started with a hands-on practical session by my colleague, I'd like to pass on the stage to over to you.
Thank you so much, Sri Gauri. Before we begin with the session, we would share with you a link to the WhatsApp group. Please join the group so that we can share the links of the activities, the videos, and also the data sets that we would be working on in the practical session today. So I'll just put the link in the chat. If the students can take a while to join the group, that would be perfect. Thank you, Sri Gauri. Sri Gauri has put it in the chat. You can join it and then just let me know once you have joined the group. All right, so we'll have a 20-minute session, and then from 1 to 2, we'll have a lunch break, and then we'll continue with the practical hands-on from 2 to 4. And for the practical hands-on, we'll share a link with you. Please join the Zoom link. We put it in the chat, and also we'll share in the group. Here is the link in the Zoom. You have to join this at 2, all right? Yes, thanks, Sri. All right, so let's begin. First of all, I'll quickly introduce myself. I'm Sonalika Ray, and I'm working as a bioinformatics project manager at Pine Biotech. And today I'll be taking you through some of the basics topics first. First of all, we'll try to clear some of the basics, and then we get a hands-on practice with the RNA-seq data, and then we'll see how we could analyze it. So we will learn today about next generation sequencing, and then we'll discuss about the different types of sequencing, techniques, and then we'll discuss about sequencers. What are the different types of data that is generated through these sequencers? And then we'll perform a practical where we extract data from these different repositories. And then we'll run 2 different pipelines to see how the data could be changed to a gene expression table, and then how from a gene expression table you can look out for the differentially expressed genes, and then look at the interesting biological pathways that the genes affect or have a role. So let's begin, first of all, by just quickly revising the basics of genomics and the Human Genome Project that we know about. So can I know in the chat how many of you have or how many of you know about the Human Genome Project? Dr. Ravi, can the students put a yes or no in the chat box?
Yes, they can put.
Okay. Yes, I can see yes. All right. And meanwhile, you are telling me about how many of you know about the Human Genome Project. Can you also quickly give me a background? How many of students are from biotechnology here? or bioinformatics or food technology, so that I know what crowd we have today and I can then alter my talk. So who all are from bioinformatics can raise their hands. Okay, you can write the name of your field as well. Bioinformatics. Who all are from biotechnology or microbiology or food technology or a different batch that I'm not mentioning right now, please write it. Biotech. Janvi has written what's that, biotech or bioinformatics? Okay, let's keep this session interactive. I don't just want to give you a lecture and go away. Let's keep it interactive. You can ask questions whatsoever you have, and let's discuss some of these recent trends so that it's beneficial to you. Biotechnology, some of them are from bachelor's and some of them are from master's. Is it like that? Okay, so most of you are from biotechnology or bioinformatics, and most of you would know what the Human Genome Project was. So quickly delving into the fascinating world of the Human Genome Project that led to the emergence of the field of bioinformatics and helped us explore the different possibilities of data analysis. So we'll quickly explore its historical context, the goals, and the significance in understanding human biology and also the diseases. We will also discuss the remarkable collaboration and the international efforts that made this project possible. So quickly going through it, first of all, let's understand how DNA works and reads its sequence, a special project that aimed at decoding the DNA that was launched. So Human Genome Project was a 13-year-long project that was initiated in 1990 and was focused on determining the DNA sequence of the entire human genome. That's what we know. The DNA sequence of the entire human genome, as scientists set out to determine the DNA sequence. The location was estimated to have 1 lakh human genomes or human genes initially. And many at that time believed that once we had the sequence, we would quickly be able to determine where all these genes were present. And this process of reading the DNA sequences and piecing them together laid the foundation for genomics. Its roots can be traced back to the time by James Watson and Francis Crick in 1953, which laid the foundation for understanding genetics and heredity, and the development of sequencing technologies in 1970s and 1980s that made the idea of mapping and sequencing the entire human genome a realistic possibility. So what was the primary goal of the Human Genome Project? It was definitely to sequence the entire human genome, which comprises approximately 3 billion base pairs of DNA. And by decoding the human genome, scientists aimed to identify and map all the genes and understand their functions. And another important objective was to develop technologies and the tools for efficient DNA sequencing and analysis, as well as establishing ethical guidelines for handling genomic information. So the completion of the Human Genome Project in 2003 marked a major milestone in scientific history, and it provided us with an invaluable resource of genetic information, enabling the researchers to gain a deeper understanding of human biology and the genetic basis of diseases. And the Human Genome Project has significantly contributed to advancements in various research fields, which include precision medicine or personalized medicine or genetic counseling, and also the development of targeted therapies for various conditions that we would be going through. So what are some of the examples of the Human Genome Project's impact? The first one is that it has helped in identifying genes that are associated with inherited disorders. It could be cystic fibrosis or Huntington's disease and many others, just to name few examples. And it has revolutionized cancer research by uncovering genetic mutations and the alterations that drive the development and the progression of tumors as well. And the understanding of the human genetic variation has provided that has been provided through this Human Genome Project has improved our understanding of population genetics and evolutionary history as well. So the Human Genome Project was a truly collaborative effort that involved scientists, researchers, and institutions from around the world, and international corporations as well. And through the data sharing that was essential as a part of the project, this project was a really success. And the Human Genome Project brought together these scientists from various disciplines, which includes genetics, molecular biology, bioinformatics, and medicine as well, fostering interdisciplinary collaboration and knowledge exchange. So one notable example is the International Human Genome Sequencing Consortium, which included scientists from around 20 research centers across 6 countries. And the collaborative effort extended beyond sequencing, encompassing data analysis, interpretation, and the development of public databases as well, to make the information freely accessible to researchers worldwide. So in conclusion, the HGP or the Human Genome Project has had a profound impact on our understanding of human biology diseases and also the field of genetics as a whole. And through this international collaboration and tireless efforts, the project achieved its goals, providing us with an extensive genetic blueprint of humanity. Now, let's quickly try to understand what next generation sequencing is all about. We'll have a quick review of the next generation sequencers and the data that we get from these sequencers, and then we'll shift to exploring data and analysis of this kind of data. So the year 2020 marks a 70-year mark since the discovery of the structure and the function of the DNA molecule. And it took almost 50 years from that time to sequence the first human genome. But soon after the technology developed, it evolved rapidly. And by 2050, the 1000 Genome Project brought a significant number of whole genome sequences to the research community. And in 2018, over 1 lakh whole genomes were sequenced. And today there is an ever increasing number of genomic, transcriptomic, proteomic, and even metagenomic data sets that are available that describe the subcellular processes with highly detailed digital data. We'll try to understand where this data is present through various databases today. And now let's quickly learn about the next generation sequencing technologies and methods. So we'll delve into the DNA sequencing techniques first, which include Sanger sequencing, next generation sequencing, and emerging technologies. We have students from bioinformatics field today in the session, and you might be aware of these sequencing techniques, but the ones from biotechnology or microbiome or food tech, this overview would be really helpful for them. So there are various sequencing methods that were opted during the Human Genome Project to sequence the data. That is a process of determining the order of nucleotide bases, which includes A, T, C, and G, in a DNA molecule. And one such method was the Sanger sequencing method that was developed by Frederick Sanger in the 1970s. That was the first widely used DNA sequencing technique. And it relies on the chain termination method and fluorescently labeled nucleotides to read the DNA sequence. So all the bioinformatics students, please tell me what are the different types of sequencing methods. One is Sanger sequencing. What is the another type of sequencing method? Please put in the chat. All right, so let's quickly understand how NGS works. So NGS, that is next generation sequencing technologies, emerged in the 2000s and has revolutionized DNA sequencing since then. So NGS enables massively parallel sequencing of millions of DNA fragments simultaneously that are significantly increasing sequencing throughput and also reducing the costs. So first here, the RNA that was extracted from the cells is converted to cDNA, and it is then sheared and placed in a flow cell. And inside the flow cell, the cDNA fragments are amplified using the bridge PCR amplification, and the flow cell is then inserted to a sequencer. And then this machine uses image analysis to capture each letter in the flow cell fragments by analyzing visual patterns, and then it converts them to a sequence of letters. And typically NGS reads are between 30 and 300 base pair long, and it includes various platforms such as Illumina, Iron Torrent, and PacBio, each with unique sequencing chemistries and applications. I can see more answers by Kushi and Ritika, pyrosequencing, exome sequencing. Good. Now let's move ahead to understand the current applications of next generation sequencing. So NGS has wide range of applications in biomedical drug designing and discovery. And some of the current applications include target identification. So these current applications could actually be a research project topics. So that's why it's very important to understand where NGS finds its applications. The first one is target identification, where NGS can be used to identify specific genes and proteins that are involved in disease, providing important targets for drug development. Next one is GWAS, or genome-wide association studies, where NGS can be used to perform large-scale genetic studies, enabling the identification of genetic variations that are associated with a disease and the development of personalized medicine. Next is drug repurposing, where NGS can be used to identify new uses for existing drugs, such as repurposing a drug that was originally designed for one disease for use in another disease. And then we have personalized medicine, where we can use NGS to create personalized treatment plans for patients based on their unique genetic profile, improving the efficacy of treatments and reducing the risk of adverse reactions. And then you have some other applications such as toxicity testing, drug development, and biomarker discovery. Biomarker discovery is one of the major fields a lot of students have been working on. And in this specific field, you're supposed to identify biomarkers which are specific genes or proteins or other biological markers that can be used to diagnose diseases or monitor disease progression or predict treatment outcomes. And in today practical hands-on session that would be from 2 to 4, we'll perform a practical where we would be looking for these specific biomarkers. Now let's look at some of the sequencing techniques that were introduced over the years. First up is Illumina sequencing, which is a widely used DNA sequencing technology that allows for high throughput and low cost sequencing of DNA. And it uses a process that is called sequencing by synthesis, where nucleotides are added to a growing strand of the DNA, and their incorporation is detected by fluorescent dyes, and the resulting data is then used to generate a sequence of DNA base pairs. And Illumina sequencing is commonly used in many areas of biology, which includes genomic research and personalized medicine. I know it's about to be a lunchtime, so we'll just quickly go over the sequencing techniques first, and then we'll take a pause. Now, there are various types of Illumina sequencers that are available. And many a times students get confused, what are these different types of sequencers? So let's quickly go over them. So Illumina is a leading provider of NGS technologies, and all of you know that, offering a range of sequencing platforms that have significantly contributed to genomic research. And let's now look at some of the notable NGS machines by Illumina. So first of all, they have the NovaSeq series, that is the latest series. The NovaSeq 6000 is Illumina's most powerful and scalable sequencing system, and it offers high throughput and flexibility, enabling a wide range of applications from whole genome sequencing to transcriptomics. And then you have HiSeq series. The HiSeq X10 is a platform designed specifically for large-scale whole genome sequencing projects, and it can sequence up to 20 whole human genomes per run, and has been instrumental in initiatives like Genomics England, 1 lakh genomes project. So these different series, basically, they are different based on the output they provide per run. And definitely one of the differences is the instrument price, and if we can sequence short reads or long reads. And then you have the MySeq series. The MySeq is a compact and versatile platform that is suitable for various applications, including targeted sequencing, amplicon sequencing, and small genome sequencing. And it offers fast turnaround times and is commonly used in research labs and clinical settings. There was Ritika answered exome sequencing, and I just mentioned one more term as targeted sequencing. So who can tell me what is the difference between exome sequencing and targeted sequencing? Ritika, want to try? I'll explain what NextSeq series does, and in the meantime, if you want to answer, you can put it in the chat. So the NextSeq series is a very flexible platform that supports a wide range of applications, including whole genome sequencing, exome sequencing, transcriptomics, and targeted resequencing. I've been using a lot of sequencing terms here. So it's very important for you to understand what these different sequencing types are. And one very interesting fact, the latest iPhone 14 camera has been used in NovaSeq cameras to take the pictures of the DNA. That is one of the very interesting facts that I got to know. Okay, Ritika is saying just heard about it. No problem, we'll go through it. So that in my next slides, I'll explain what exome sequencing is, what targeted sequencing is. So for now, I'll take a pause and not go over that topic right now. Next is nanopore sequencing. So when you talk about sequencing techniques, there are three types of sequencing that I'll go through. Illumina, nanopore, and PacBio. Khushi is saying, I guess exome sequencing refers to the, yes, absolutely correct. So we call exome sequencing. So there are three types of sequencing. Genome sequencing is when you sequence the entire genome. And then when you sequence only the exonic regions of the genome, that is exome sequencing. And when you are targeting a specific gene or a specific portion of the genome, that is targeted sequencing. Very easy. Next up is nanopore sequencing. We discussed about Illumina. Now let's see what nanopore sequencing does. So as the term says, nanopore. Basically what happens in a nanopore sequencing, it works by passing a single strand of DNA through a protein pore. So this is a pore here between the protein, and it passes a single strand of DNA through a protein pore, and with each nucleotide causing a change in the ionic current as it passes through. These changes are detected and used to generate a sequence of the DNA molecule. And the key advantage of the nanopore sequencing is that it can generate long read lengths up to several kilobases in real time, allowing for detection of complex genomic features such as structural variants and repetitive elements. And it also has the potential to be portable, that is used in field-based applications such as pathogen detection. Nanopore sequencing has emerged as a very promising alternative to traditional high-throughput sequencing technology such as Illumina. Now, it's not that because of nanopore, Illumina sequences are not used anymore. They are definitely used for different experiments. Different types of sequencers are required. One of the drawbacks of Illumina sequencing as of now is that they can't sequence long reads. They just take short reads of DNA to sequence. But Illumina would soon launch some sequencers where it can really sequence the long reads, just nullifying the limitation. Now, what are these different types of nanopore sequencers? I just mentioned that they're portable sequencers. See how small they are in size. They can actually be carried very easily. So the nanopore sequencers by Oxford Nanopore Technologies, they use nanopores to directly read the DNA sequences, as I just mentioned, and they offer real-time analysis, and they are portable sequencing devices. So they're very interesting to read about. Now, nanopore sequencers, they basically are of three different types that I'll discuss here. One is MinION, another one is GridION, and then you also have the ProMethION. So the MinION is a very portable and compact sequencer, okay, as you can see here. And it uses nanopore technology for real-time sequencing. And it allows users to perform sequencing experiments in various environments, which can include remote locations or in the field. And then you have a grid ion, that is a high throughput nanopore sequencer that is designed for large scale sequencing projects, and it offers increased capacity and also parallelization, enabling researchers to process multiple samples simultaneously. And then you have ProMethion, that is the most powerful nanopore sequencer that is offered by Oxford Nanopore Technologies. And it is designed for ultra high throughput sequencing that allows researchers to generate large amounts of sequencing data. Now, as I mentioned, nanopore sequencing has gained attention for its long read capability, real-time analysis, and portability as well. And it provides advantages in detecting various types of genomic variations, such as structural variations and epigenetic modifications as well. And these features make nanopore sequencers appealing for researchers who require rapid and accessible sequencing solutions. And next, I'll discuss PacBio with you. PacBio is a single molecule real-time DNA sequencing technology that uses polymerase enzymes to synthesize long strands of DNA. And these long strands of DNA that are synthesized are then imaged as they pass through a zero mode waveguide, and the sequencing process generates long reads with average read lengths typically ranging from 5 to 15 kilobases, which allows for detection of complex genomic features such as structural variant, repeated regions, and epigenetic modifications. Now, there are two main types of PacBio sequencers. One is the Sequel-1 system, and another is the Sequel-2 system. And the Sequel system is basically a popular PacBio sequencer, the Sequel-1 system, that provides long-read sequencing capabilities, and it offers high throughput and scalability, making it suitable for a wide range of applications. And then you have the Sequel-2 system, that is an upgraded version of Sequel-1 system, offering improved sequencing chemistry and data.
हो रहा है
Now, I want,
नेटवर्क का इशू होता
sorry, sir. Am I audible? Shri Gauri?
Yes, yes,
Yes,
yes.
sir.
One more request to you, Dr.
Yes.
Sonalika,
We
that
have reached the time
we
limit.
will conclude first session so that they have lunch, participant, and then rejoin at 2 p.m. with the
Yes.
same link. With the same link.
Okay, with the same link, sir. All right. So, yes, they can rejoin with the same link, and I'll share in the group some activities. So once you're done with your lunch, you can then run these activities where you will learn about DNA microarray sequencing and also sequencing by synthesis. So I put it in the group. You can try out these activities. And those who have not joined the group yet, they can join the group in the meantime, because all the important information would be shared here, and I don't want you to miss it out. So we have right now just 7 participants, but I can see more students here. Thank you, Shrikari. We have shared the link of the group once again. Please join the group in the meantime, and let's catch up at 2 p.m. We'll quickly complete the basics, and then we'll start with the practical hands-on for data extraction and data analysis. So do you want me to just have this meeting on, or shall we leave the meeting and rejoin at 2?
Your choice. I mean, you can keep it joined or you can rejoin. So there's not an issue. This session will be on.
All right.
during this period. And dear all participants, after lunch, please rejoin at 2 p.m. And we have many things to do in the second session too. Right. Thank you.
Thank you, sir.